I, I thank you all for being here, and particularly also the speakers and those that came from very far to be here with us. And I have a special thanks to all the staff at ICTP and the Principia Institute. Without their help, that couldn't be possible. And they were extremely patient. They had all the support and all the help. So there is a big staff involved. Besides Nathan, who embarked on this idea, Elisa, Wellington, William, Thiago, Umberto. So they have a fantastic group that helped us to have this to occur. For some of those here don't know the story, the idea how this meeting started, it was like 10 years ago that Paul was going to get married here in São Paulo, and he invited all his friends from academia, and there was a large crowd, and they say, well, do we have a meeting or something like that so we can have an excuse to come? And there would be no way that we could accommodate almost 30 people. And so at the last minute, okay, well, so we have to organize a meeting. And so this is how it started. And it was a fantastic meeting because it was just like friends getting together, and was a fantastic, very fruitful meeting. And as Hopfield once wrote to José, that science is a social enterprise, and it's so nice when that's done among friends. And so it's so nice to have friends from long time and friends to be. So we are all welcome, and that's it. And we have a few. Just a brief videos explaining what is the ICTP and what is the Principia Institute. So please, can we have the films, please?
O Instituto Principia é um centro de produção e difusão do conhecimento científico e de conexão da ciência com a sociedade. Com atividades científicas desde 1951, tem atualmente 3 programas de atuação relacionados à pesquisa, educação e divulgação científica. No programa de pesquisa, acontecem eventos internacionais sobre temas atuais para diversas áreas da ciência. No programa de educação, a Escola de Talentos oferece oportunidades de desenvolvimento pessoal e acadêmico a jovens que tenham grande interesse e altas habilidades para a ciência, oferecendo uma tutoria científica com pesquisadores renomados. No programa de divulgação científica, existem diversas atividades com ênfase para o público jovem, como o You Science, o cinema científico e publicações em várias redes sociais. Conheça, participe, amplie seus horizontes através da ciência. www.institutoprincipia.org.
So this is, you got a picture about this institute, and as you've seen, they have this, one of the areas of interest of the institute is biological physics. So especially for the students for the next year, there are already some schools planned, and with this fantastic welcome we had from them. So we plan to have other meetings such as this one in a near future. So we keep an eye on that. Before we start our lecture today, I just want to give a few comments on things nearby for the students. And well, there are foods that you can find both in one corner when you go down the hill, and also in the other direction they have like two restaurants side by side. So and they also have the restaurant right here, the big old house. So during the lunchtime, you just go around and find whatever pleases you most. Regarding those that have a poster to present, apparently we don't have that many, so probably those who are going to present a poster, they can have that at their site for the entire meeting, and that's going to be fine. And we encourage all the students, exactly, this is a fine opportunity to get in contact with all the speakers, etc., and try to interact with everybody. So that's the idea of the meeting. So we also have, this has been broadcasted by, people have access to Zoom as well. I don't know if you have friends that couldn't come, they can access at the web page of the meeting. They have the instructions on Zoom for those who can't come. And so with that, we can go to start our program itself. And today is a really pleasure to have with us Peter Wolynes. He came already for the SBBF meeting, for those who were here. Well, it's a big honor to have him. It's his first time in Brazil, and he's a legend, as I said, in the field of molecular biophysics, and has worked in many areas. And today he's going to talk about a different topic than what he did at SBBF. Peter has been in several places, started in Indiana, went to Harvard, and Urbana, Illinois, San Diego, and now at Rice University. And for smaller periods have been everywhere, Japan, France, you name it, so many other places. And so today, Peter, please, that's your stage. Thank you so much.
Yeah. Hello. I feel a little bit like Carl Sagan now in this format. We'll try our best. Okay, remind me. That's good, because I'll just say movies. You think this thing doesn't work now? Okay, good.
It will go back and forth, and they will control it.
Okay. Well, it's a great honor and a great pleasure to be able to speak here again in Brazil. It's been a very nice visit so far, and of course, as I mentioned previously, I have had a long sort of connection with the Brazilian science establishment through José Jonacek, and it's really nice to see so many of you here, while previously I've seen you in San Diego and Houston and so on. The topic I'm going to talk about is something that's fairly new for me, the last maybe work of the last 5 or 6 years. And it's kind of odd that I got here because, in fact, the title reminded me of something that happened more than 50 years ago when I was an undergraduate at Indiana University. In my last year, I was doing theoretical chemistry research. I'd come from trying to be an organic chemist, and I started to do things about the kinetics of organic chemical reactions. And the people said, oh, the organic chemist said, go please talk to these theory people. We don't care about that stuff. And so I got connected with the theory people there, was very interested in the dynamics of how chemical reactions went on. And I had a sort of an advisor named Bob Roberts, who was in chemical dynamics. Now there were 2 other people in theory there at Indiana. One was Don McQuarrie. You probably know the textbook of McQuarrie's Statistical Mechanics. And another guy was called J.S. Griffiths. He was famous for his work on crystal field theory, but he was also the person who had figured out or came up with the idea of the prion, which later won Prusiner the Nobel Prize. And both Griffiths was working on that problem, but also on the brain, and McQuarrie was interested in the brain. And I remember saying to my advisor, I said, oh gosh, I don't really like this biology stuff. You know, I really like this, I really like, you know, real science and things like dynamics. And he said, oh, Peter, you just shouldn't care about what something is called. And you know, you should work on whatever you want to work on, and if it's biology, that's fine. And I didn't really follow that advice for very much at all. Maybe about 5 or 6 years later, I wrote my first paper. Not 5 or 6, well, 5 years later, wrote my first paper that had some biological molecules in it. But that was kind of a hobby. And then later it became a serious part of my work when I turned to thinking about protein folding and interacted with Hans Frauenfelder. So I never really did anything with the brain. And then in about 5 years ago, several different projects that we were doing in protein dynamics sort of led us naturally to questions that were fairly deep about how the brain works. And I'm going to give you sort of a flavor of some of these ideas that we've been playing with. I think some of my reticence to deal with the brain was completely justified. I think when I compare the experimental situation that I encounter in looking at neurobiology compared to what we encounter in molecular biology, it's just worlds apart. Almost everything is speculative. Almost no measurement is very easy to do. You know, just to even do an experiment on a mouse requires tons of paperwork in the United States. And if you think about it, almost anything neurobiological has an intrinsic timescale that's so long that it means you can't do many experiments. So this is why it's a very difficult subject experimentally, and why I feel also that you should be very careful about things I'm going to be telling you as experimental facts, because I'm not sure in my own mind whether I'm telling you them correctly or even whether the field understands them correctly. So in terms of reminiscences, of course, the most famous example in our Western culture of reminiscences is this famous quote from In Search of Lost Time, à la recherche à la temps perdu, or something like that, by Marcel Proust. And he tells this one incident, which has in some sense been taken as the touchstone of all of neurobiology. He says, no sooner had the warm liquid with the crumbs touched my palate, this is of a certain pastry that he's eating, a madeleine, then a shudder went through me. So something physical went through him at that point. And I stopped, intent upon this extraordinary thing that was happening to me. The exquisite pleasure had invaded my senses. Something included detached, and at once the vicissitudes of life had become indifferent to me. Where did it come from, this association, we would say, between the madeleine and this great warm feeling? He says, and suddenly the memory revealed itself. I'm having trouble reading it with my glasses from this distance, but anyway, he says, the little piece reminded him of a Madeleine which he had in his Aunt Leonie's home. So this thing, one sort of sensory input brings to mind all this other stuff and all these other actual actions and other reminiscences of these things that were past. So the real problem is how our memory is formed. And as I'm saying, we'll be talking about some work on this problem, and it has some fairly deep aspects to it. At a very crude level, there's been an understanding that there's something like a reinforcement learning in the language of the psychologists. But it's something going on in the biology and the physics of cells, not at the whole level of whole organisms. So the idea is that, somehow or other, when a neural pathway is stimulated, say you have a signal come in, and it causes one particular neuron, let's say in your taste bud, to do something, then it will go and start off interactions with all kinds of other neurons. If you take another bite, you get another, that same feeling, the same neurons. And so all of a sudden you get this idea of Madeleine always gives the same neuron going off, connecting to some other neuron, going to like salivation of your, as you eat. So there's this connection between neurons which is reinforced by some mechanism. And the mechanism is believed to be something along the following lines, that when a neuron is stimulated, if it's repetitively stimulated at the right frequencies, it causes a change in the connection to the other neuron. The connection is called a synapse. And that change is to basically enlarge it. That if a neuron gets used a lot, it grows a lot. So that connection means that the next time you get even a tiny bit of Madeleine, you immediately think of the same things and start to salivate. And microscopically then, it is at least clear that something like that is what initiates the formation of a memory. And I think if I can get, there is a movie here, but I'm not sure if it runs. Can you see, sir, whether you can get the movie to run? Ah, super. So what you see is that this region there is being stimulated by an optical signal, actually, to release calcium. The release of calcium causes an electrical discharge, as well as a chemical discharge of neurotransmitters. And then you see that it goes up against this other little knob that goes off of that. That's called a dendritic spine. And that dendritic spine, after it's been hit once, it actually grows. So that means the next time you don't even need quite as much of an electrical hit for this to happen. I've really oversimplified there because it has not only to do with the strength of the hit, but the frequencies of the hit and so on. There's a lot of complexity of processing of this electrical signal. It's not just strength, it's frequency of discharging, and it's rather complicated. So even an individual dendritic spine, an individual neural cell stimulating it, is a very, very complicated thing, and that's what we're going to talk a bit about. But it's basically that somehow or other, electrical signals cause the dendritic spine to grow. And it's been found that the electrical signal that's most important in the end is an influx of calcium. You can see that the idea is that somehow calcium comes in, there's some structure holding the dendritic spine in its own shape. By calcium coming in, it rearranges. So first of all, this is very interesting from the point of view of soft matter physics. There's a lot of interesting soft matter physics of how does a dendritic spine have a shape when it's really made up of these kind of like tumbleweeds that are holding the cell apart. So part of this is that there's an enzyme called CAM kinase, calcium calmodulin dependent kinase 2. And this enzyme is something that phosphorylates things. That's what kinases always do. And this phosphorylation is responsive to calcium. That's one thing that happens. It starts to phosphorylate all kinds of things. But also, even though this is an enzyme, it's present at a very high concentration. On a mole basis, there's about 10 actin proteins, actin monomers, for every CAM kinase. So it's really kind of a structural element, as well as being or it has the potential to be a structural element, as well as being a kinase that does catalysis. Let's see. So the idea of this then is it's something that can form a structure with actin, which can be stable most of the time. And then when calcium comes in, that structure can rearrange. Of course, actin is already a dynamic thing that grows and shrinks and so on. I think we might be hearing about that from Garrick Papoyan somewhere. I haven't seen him yet today, but hopefully he'll be telling you about that in this session, in these talks. Okay, so here I think it's just the slide I should have gone to that explains there's this remodeling caused by the calcium signal. So our work has been to try to understand the structure of this complex, this set of complexes between CAM kinase molecules and actin, trying to understand how it could be regulated and so on. Now, probably the appropriate approach to this would be an experimental one where we try to go in and find the structure of this dendritic spine. But the dendritic spine is very, very small, and everything that's holding together is not particularly strong. So it's kind of hard to be sure what its structure is by direct observation. And in fact, I've tried to get experimentalists, well, first of all, I got interested in this problem partly from experimentalists, from Neil Waxham, who had already been collaborating with Margaret Chung, one of José's ex-students. They'd been interested in this calmodulin part of the problem. And I tried to get this guy to do more experiments on the structures, and it was already so hard that he was willing to do theory with us, or help us with theory. So one of the things is, of course, our understanding of molecular structure has really advanced tremendously. Even though you hear a great deal about AlphaFold solves the problem of protein folding. In fact, we had solved the problem of protein folding long ago, and we had at least decent ways of doing predictions of structures and trying to understand the association of different molecules with each other, although albeit they always took quite a bit of work, and there's also the possibility of making errors, and that's of course important. So anyway, one of the tools that we've been developing for almost 40 years now, 35 years, is the use of neural networks, but in a different way, neural networks to inform the learning of an energy function so you could simulate the dynamics of association of different proteins with each other, as well as the folding of proteins. So the representation that we use here is a sort of coarse-grained one. We don't show every atom, but we show enough atoms that we can sort of reproduce the backbone structures of most proteins. We have usually one individual or maybe one or two side chains to represent the character of the molecule. And this is an approach we started in the late 80s. And it's sort of reached maturity, you can see, somewhere around 2012. So this is the kind of quality of structure prediction one could do in 2012 using this kind of model. So this gave us some, you can see that we superimpose, actually this is somewhat earlier than that, I now see, because things are represented in the colors of UCSD. The correct structure is shown in blue, and the incorrect structure, so to speak, the predicted structure is in yellow. You can see they mostly are on top of each other. So low resolution structures are reasonably well learned this way. And actually, one of the surprises of our recent work with this is even thermodynamics is not terrible that we obtain from these sort of energy functions. It's not as good as structures, but it's still pretty decent. And then this is the kind of sort of mature form of the tool is described at the bottom. This is work of Aram Davtyan, Nick Schaefer, who was started with me in UCSD, moved on to moved on to it with us to Houston. And then also we involved Cecilia Clementi and Garrick Papoyan, who really was the person who discovered the interactions that are most critical for this, the so-called water-mediated interactions. That's what the name comes from, associative memory. It's neural network-based, water-mediated structure and energy models. So what is the structure of the molecule itself, the CAM kinase? Well, part of the molecule structure had already been determined. The part that was involved in the enzyme is this sort of hub of 6 monomer units. It's really a rather large structure. That's called the kinase domain. That's the thing that does all this phosphorylation stuff. And then it has a couple of other domains. One's a linker domain that's supposed to be intrinsically disordered. I always point out that Jose gets upset when anyone says intrinsically disordered, but over the last 20 years he gets less and less upset. But it's at least not ordered. And then there's a thing called the association domain. None of those parts, other than the blue kinase domain that were shown there, had a crystal structure because they were intrinsically disordered. So part of the molecule we sort of understand, the biggest part of the molecule, and it looks like this hub. My student Ming-Chen Chen used some natural intelligence to figure out where the hub molecule might bind to actin. And the reason he did that is if you look through all the structures of actin binding proteins, you find a fairly natural binding pocket where almost all the things that bind in a very specific way to actin that show up in your X-ray crystal structure bind somewhere near there. So that gives you some guidance. So now you can do a molecular dynamics simulation with the awesome Hamiltonian with some guidance towards that general area. So with this minimal construct, just the single monomer of the kinase coming up to an actin, you're able to do a simulation to see if it actually can stably bind in that location. And I think there's a movie here that could be played. Sir? It's not the best movie, but could someone, well, I'll go on. This is, okay. So you can see that there's a sort of general push towards that region. We, of course, pointed the molecule near to where it could bind, and you can see that it binds, and one ends up generating quite a few structures this way, and you can look at the consensus structures and see which ones are the most stable and so on. So you get this sort of picture, and that can be compared to a cryo-EM investigation that was done by Neil Waxham. And you can see various features of this are at least roughly right, where the thing goes and fits there. But in addition, just from this part, maybe I have to go this way, you can start to see what would happen if I had many CAM kinases. So for example, the actin fiber structure is itself known. This sort of unit, if it's going to bind, has to bind in a way that's sort of a helix. Everything's always a helix. It's a mathematical thing. That's something that has the same laws that are applied uniformly along something in a sequence ends up to be a helix. It'll be a helix. Maybe it becomes a helix of helices or helices of helices of helices. But this is why it was possible for Pauling to figure out that the specific alpha helical structures and so on, and how Watson and Crick could figure out DNA structure. If you have a constant rule, and you have to have that in a fiber, like the actin fiber, then you'll end up with a helix. And you also can get junctions. You can get junctions where these things fit, not just trying to bring things together all lined up, aligned with each other, bundling, but they'll also do possibly have some joints, have regions that will become joints between them. So the kinase could provide all this sort of rigid geometry that you need, and it fits the cryo-EM reasonably well. But what do these other parts of the molecule do? They're intrinsically disordered. We could, of course, just try to run awesome on them. But in this case, we also wanted to understand whether those regions could bind to the actin by themselves. And there's an experimental way to do that, which is to just cut the sequence up of this region and make small peptide fragments. Looking at those small peptide fragments, you can then pour actin over it and see where the actin will bind. And in this way, you can figure out which segments might be binding and which segments might not be binding. In a sense, that's the same thing that the associative memory part of a Hamiltonian does. But when you have an intrinsically disordered protein, you don't have any memories, that is to say, a database of known intrinsically disordered things. So from this, and then combining that with awesome simulations to use the guidance from those, one can end up with, a structure in which not only does the catalytic domain have the option of binding, but the regulatory domain and the linker and the association domain can be placed onto the same structure. And we can sort of compare where they're predicted to be with the strength of the binding that we have in this experiment. By the way, this experiment costs about several $1,000s to buy the plates. And in this case, Neil was willing to pay for the plates. For another protein that I'm not going to be talking about today, he was unwilling to do the experiment until I paid for the plates. So as I understand it, the rule is that if you pay for the experiment, you've become an experimentalist. But anyway. So multiple domains are connected. And now one can start to sort of see how the whole thing could come together. So we started to do also awesome MD with no bias to a specific region and see if we would get the same kind of results we would get with those biases. And I think there's a movie here, or maybe it will go forward if I just press the button. Oh, if I go back, yeah. So here we're just taking those regulatory regions and seeing where they fit. And we see that they actually sort of fill in places between the places where the large hub domain could go. So it ends up looking something like this as the structure. And again, we can run a movie of what the motion looks like, I think. Let's see. Yeah. So in the whole thing, you have some parts, there's some part that still remains disordered, but the other parts lay down very nicely onto the domain. Well, as I said, one of the nice things about awesome is it gives you at least some idea of thermodynamics of association. So in this case, we can start to look at how hard is it to pull the CAM kinase off of the actin. And we can see that there's going to be three things that contribute. There'll be the binding energy of the hub domain, the binding energy of the regulatory domain, and of this association domain. And we can separately monitor the free energy profile of pulling one of those things off. So we can make an estimate of the binding capacity. What you could see is there's actually a relatively small binding from these regulatory domains. I mean, small in the sense of a few kilocalories, but that's all that we need basically in biology, especially if we can regulate that. If we can change something from binding with by 3.8 kilocalories, that's a factor of 10 to the fourth in the binding constant, let's say. No, wrong. No, right. It's a little bit less than that, but a factor of 1,000 or so anyway. So that means that these regulatory domains can take the binding from being very strong until, if they were removed, to being fairly weak. Why would they be weak? Well, once you've added, or how could you make them weak? Well, that's actually what the calcium signal does. So the calcium signal comes in, it makes calmodulin be activated, and then what we see is that calmodulin can compete for the same binding surface that's used in the regulatory domain. The regulatory domain can combine with the calmodulin, or it can bind to the actin. So this means that you can kinetically strip the molecule off. So this is a process that with Betsy Komovets and I called molecular stripping, and we heard the other day from Ben Schuler about another example of molecular stripping. It's a way that you can use intrinsically disordered things to do things with some degree of kinetic control. So this story is a story that sort of explains how these interactions of somewhat disordered proteins can either allow the formation of a large structure or cause the dissolution of that structure. So what apparently happens is when the calcium comes in, the calmodulin gets turned on, it now competes, strips off the CAM kinase, which is already there, and now the actin cytoskeleton can continue to do its work and move and expand if it wants to. Okay. Let's go on. So there was actually a puzzle that I had gotten into before this, again by accident, in studying protein dynamics. And it goes back to this prion problem that I mentioned with J.S. Griffith's idea. You've probably heard a lot about prions as being the causes of various diseases, like famously Kuru in New Guinea and mad cow disease. And so they're always thought of as bad things. But there's been at least one proposal that it's actually the storage of information in prion structures, that gives you the long-term memory. And this was a question that was brought out first by Francis Crick, or fairly clearly by Francis Crick, that you're storing memories from 50 years ago, right? I told you about something happened more than 50 years ago, and yet the lifetime of most of your proteins is only a few minutes. Of course, there's proteins in your eye that last longer than that, but they form crystalline structures and so on. But in general, you have to have some very, very large organized object to store information for long times. So Crick actually proposed that prions, what we now call prions, they hadn't been discovered quite yet, that some kind of aggregates of protein was one way to store memory. The other way he proposed was actually phosphorylation patterns. So it's interesting that CAM kinase is involved in phosphorylation. So there's still mysteries here, but he postulated these were the two ways one could overcome the problem of turnover. And there was a specific protein that was implicated as possibly being involved in this. It's a protein that was studied by Eric Kandel, who's a famous, actually originally he was a psychoanalyst, but he became a neurobiologist and won the Nobel Prize for his work in neurobiology, and Susan Lindquist, who was a prion specialist at MIT before she passed away. And they found in a variety of animals that they could study in the laboratory that there were some proteins that formed aggregates, and they did so only when memories were formed. So they studied this snail called Aplysia. They also did experiments on mice, where they would have mice, they would throw them into a swimming pool and see if they could remember where the door was to get out. It's kind of a little bit of a mean experiment in my view, but in any case, they were able to monitor the memory of these animals. They're also able to manipulate their genes so that they could either have the protein that forms this prion or not have it. And so from these sort of studies, they came up with the idea that when you have a long-term memory form, that actually that this is stored in the formation of a prion of this CPEB. There's also interesting questions. Why does having an aggregate do something? Well, one of the reasons is CPEB in the aggregated form acts as something to improve the translation of cytoskeletal proteins. That's another part of the story that Xinyu Gu worked on in my group, but I won't be telling you about it today. But I think we can understand why changing the structure of the protein may allow it to act in a different way and be a translation organizing element. But anyway, I got into this particular protein because I asked my student Ming-Chen Chen, he's joining the group. I always gave people in those days a simple problem to just see if they could run the prediction software. I said, predict the structure of CPEB, because I give them a problem of a protein that if we get it wrong, I don't care. And if they get it right, then there's something interesting. So here he did it. And what he found was that CPEB naturally aggregated, but it aggregated into helical bundles. So at first you go, oh, this is wrong. But no, the answer is the experiments actually show that CPEB on its own normally just aggregates not to form something like a prion, but instead forms a helical bundle. And the structures are more or less what we got. Well, they don't know the structures, but they're consistent with what we got. However, you can see we've got dimer, trimer, and the larger structure. If I can go on. So, of course, when something's coiled up into a helix, it's relatively short. But in a prion is a beta sheet-like aggregate, and it's still a helix. The beta strand is itself kind of a helix. It's repetitive, but it's a long one. Well, how do you make something long? You pull on it, right? And so Ming Chen said, let's go ahead and exert force. And in fact, one was able to calculate how much force would it take to take one of these helical prion aggregates and make it turn into instead a beta sheet-like aggregate. And the amount of force it turns out to be needed is only the amount of force that's easily exerted by the cytoskeleton. And so actually one of the problems with continuing this in the lab is it's very hard to make CPEB aggregate on its own. You can only make it aggregate by doing something like pulling on it and doing it in some combination with actin. So it's a protein that's hard to make into a prion, but it does make a prion when you pull on it. This is also good for the people who do folding. Often people say, oh, all proteins aggregate and form prions. No, that's not true. This one you've got to work. And as we said, the amount of force that you can calculate, and because the thermodynamics can be worked out, is just about what you need. And you find that CPEB3 also binds to cytoskeletal proteins. And the proteins that are turned on in their translation when CPEB is in its prion form are also cytoskeletal proteins. So you can form a kind of closed loop, partly closed loop again in the signaling that says that you rearrange the cytoskeleton by the CAM kinase. It starts to move. It pulls on any CPEB which is bound to it and pulls on it and makes it form a prion. The prion that's formed now says, oh, let me go and bring in and recruit other proteins that are needed for making cytoskeletons. And so this is one way you might close the loop here. Okay. So as I said, in studying aggregation, these are sort of positive example. It's a positive example of aggregation. We also have studied many other examples of aggregation where it can happen with greater spontaneity without having been caused by the cytoskeleton. And it's always been a sort of a surprise to me that almost all these, that's a bad way to say it. Many of the most famous aggregation diseases are diseases involved in neurodegenerative disease. There actually are other things like type 2 diabetes and so on is caused by aggregation, but is in your pancreas and not in your brain. But the ones where anyway we notice it a lot are things where it happens in the brain. And it's always bothered me, and it still bothers me because we don't have an answer to this, that we actually have almost no idea of the mechanism. So that you have, for example, people who say amyloid beta protein causes Alzheimer's disease. You have other people who say tau protein, when it aggregates, causes Alzheimer's disease. And this makes it actually a problem to write proposals in this area. Whenever you write a proposal here, you'll get someone on the panel who says, no, I know your protein is not the one. And then you say, but I've also studied the other protein. And they say, but that's in your other proposal. And then that proposal someone else complains about. But anyway, we've done a lot of studies on A beta. And we've done quite a few studies on tau. But I'm going to talk about a protein that's I think even more strongly implicated in neurodegenerative disease. That is the protein involving Huntington's disease. Now, the person that I'm showing you here is Nancy Wexler. I ran into her when I was on several committees. Sometimes it's worth being on committees. You meet interesting people. Well, there are two things relevant. She's actually the person who discovered the Huntington gene. She did that because her father had Huntington's disease, and he was also very wealthy. So he started a whole institute to study Huntington's disease, and she was able to find this particular gene. And it was found by investigating a group of people in Venezuela who are very highly inbred, and so they have very different forms of this Huntington gene, but they're all the same, and it's something where the people get the disease at quite early ages. That's again one of the problems of studying diseases of old age. It's hard to get graduate students who are willing to wait 60 years to see whether that graduate student, see whether his advisor got Alzheimer's or not. Okay, but anyway, Nancy is here, and actually, it's a little bit sad. She was the one who discovered this. She also, it does turn out, does have Huntington's disease. So this is several years ago, and she's not been doing that well. But it's a sad story. So here is what Huntington's disease is caused by this protein called huntingtin. And the main part of huntingtin is one of these very, very simple proteins. It's a homopolymer of glutamine. It's QQ, 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 QQ. Okay. And what happens is in reproduction, sometimes in duplicating a gene, you find that it starts to stutter. And so I think the sequence for Q is CAG. So you sometimes get CAG, 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 and then sometimes in the next generation, you get somebody who has CAG, 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 and then it keeps doing this. So first of all, just to make you all feel good, all of you have Huntington with lots of repeats in it. Okay, but the problem is that it's when you get a lot of repeats that you start to get the disease at a very early age. So this is a plot of the age of onset of Huntington's disease against the length of the repeats. And you can see the people who are out in Venezuela, there are not very many of them, but they get this disease maybe at age 15. Someone who has a protein of length, I'm trying to read what the number is here, 40 or so, you start to get the disease. If you have something less than 40, which most of you hopefully have, less than 40, then this is not a disease to worry about. However, in every generation, this typically gets longer. And also another thing that might be worth knowing is seems to pass, this stuttering problem really occurs in the formation of the spermatozoan. So the difficulty is, this is a good reason for women to marry younger men. Okay, don't go with those real old guys. Over generations, it creates problems in Huntington's disease. Okay, so what happens in Huntington's disease is you form some aggregates. Some of these aggregates are sort of more or less just inclusion bodies are fairly amorphous, but then they can, when you have the long form, they then end up being these sort of crystallized, sort of prionic aggregates. The question then is, what's different? How does length change this? Well, what we found was that even polyQ by itself, its structure depends on its length. If the length is rather short, it typically is just quite disordered. But you can see that if there's some attraction between the Qs, which is what eventually gives rise to the formation of an aggregate, if an aggregate gets long enough, it can just close on itself. So we find that, say, at length somewhere around 24 to 28, you have a switchover from a molecule that's not folded over to one that, not folded over to one that folds over. And so we would argue that this is, and we'll see in a moment, that this is why there's a length dependence to the global aggregation, this change of the monomer structure. So let me go ahead. This has also been studied experimentally. Huntingtin itself, that's the protein that causes Huntington's disease, is not actually pure poly-Q. It has an N-terminal fragment, and it also has a C-terminal fragment, which is about the same in all of these cases. So it's been studied by Wenzel as various lengths of these peptides. And what one finds is, in fact, that the short lengths have to be, you have to have a high concentration for them to nucleate, and the longer lengths seem to have a lower concentration to nucleate, as well as the nucleation cluster is quite a bit smaller when the length is there. So we actually have data on the solubilities and kinetics of aggregation of these things. And we can do simulations of the aggregation. I think we can play this movie. Let's play it once. This is the simulation of Q20. And you can kind of see all the Q20s are just hanging out there. This is at a very, very high concentration. One of the problems is to actually study the real process, you should go to a very low concentration, but that means molecules have a long time to go around. So actually to turn this into a quantitative theory, you need to have a correction for the concentration, but that turns out to be a very interesting piece of statistical mechanics using the grand canonical ensemble. But you can see here it eventually nucleates, and one can see the nucleation process. If I go on to the next slide, so here we can get an aggregation free energy profile. We can get it at the simulation concentration. We can simply count how many guys we have that are monomers, how many dimers, trimers, etc. And of course, the logarithm of the count is basically a way, with a correction factor, is a way of figuring out the free energy profile as a function of the number of contacts. This is actually one of the things that's always been a pet peeve of mine, that people who study amyloids often put the energy landscape of the amyloid on the same thing as the monomer. But it's a completely different system. And amyloid has a variable number of particles, so you always have to have a variable like the length that's involved. And statistical mechanics, and this is the analog of those free energy profiles we see all the time in protein folding, but we have it now with aggregation. And then we can also see that this predicts what the nucleus size is for Q20, and you need at least 3 of these guys to get together to nucleate at reasonable concentration. So let's move on to the next slide. Okay, now for Q30, you'll see that the Q30, already the guys flop over on each other. When the two things have flopped over on each other, they're now very rigid. So when they take 2 rigid guys and bring them together, they find it very easy to stick. They form lots and lots of contacts. They're no longer so disordered, they now have partly ordered. So you have less entropy loss, and so that's the main reason why the greater length guy, by doing this initial process of nucleation, internal nucleation, it's gone one step further towards forming the aggregate immediately. And if we move on to the next slide, we have the numbers there. Hello? As I said, we're doing this not just for the those are movies with the pure polyglutamine, but now we can look at Huntingtin itself. The whole molecule has lots and lots of residues, but the part that's involved in aggregation is relatively small. I've forgotten the size, but it's a relatively small thing. It's about 17 plus another 25 on the other side. So again, we can simulate real Huntingtin. And what we find is a few interesting things. The N-terminus actually forms one of these helical bundle structures like we were talking about for the other prion. And they act to facilitate aggregation. If you just had this N-terminal fragment, everybody would have Huntington's disease, practically, even at Q20. And then the next slide, we see, though, that, can I go back? How do I go back? Okay. That the C-terminal does the opposite. The C-terminal, which is made of prolines, prolines are very hard to put together into an organized structure because they have this bizarre sort of internal bonding. And so polyproline part inhibits their aggregation. So this is why we don't all have Huntington's disease. And if I go on to the next slide, we can estimate the concentration at which you switch over from being soluble to being aggregated. And we can see that you, at the length scale where the disease starts to be seen, the Q contacts start to be seen, then we get to a point where the concentration within those within those amorphous bodies is enough to cause the aggregation into the ordered one. So in a sense, this explains why there's a length dependence to the onset of the disease. It even gives quantitatively a reasonable number for where that is. So I think that's about it. All these topics were things that we've been studying. We partly had a separate grant from the NSF to explore this on dendritic spine mechanobiology. But the rest of the work has been funded as part of the CTBP, and we have a group of people which we call the memory group, which involved Margaret, who used to be at our center and has moved off to Washington, and Neil. With Herbie, we're interested in questions of the mechanics of the cytoskeleton, which is another part of this problem, obviously. But at the molecular level, these are some of the stories we've been able to find. Thank you.
We have time for discussion. Questions?
Yeah, yes, Vittore. Yeah, or.
In regards to the last story about the Huntington disease, I've been listening to talk which try to highlight the protective role of disaggregates instead of the fact that this might cause the disease. And another thing that might be interesting, maybe you can comment on it, is that maybe the same mechanism that form aggregate that can increase the interaction with other molecules.
Oh, I'm sure that's involved. I mean, so first of all, on the first one, well, there's several things that could be said. I think here the case for this being involved in the pathology, in the negative pathology, is, I would say, pretty strong given the numerical agreement of this mechanism of length and the sort of monotonic dependence of length of the age of onset. I would say that's why this is my favorite example, so to speak, of a biophysical disease. When we come to A beta, we have all kinds of questions. So some people say that in fact aggregates aren't that bad. And there are people, everyone has aggregates at say age 65, but not everyone has Alzheimer's. And like I said, just because we don't, and so there's been, I would say, a kind of desperation move, which is the desperation move is to say aggregates are a problem because it's actually some small oligomer, which perhaps also has one structure that would maybe be form a hole in a membrane. That's the most popular sort of thing. But I would say there's not a lot of evidence for that. It's something that people say. And I think one of the problems is that all these proteins are also have many of these proteins have functions, but we don't know what they are. So A beta protein in its normal form, we have no idea what it's doing there. Tau protein does interact with microtubules, so it does get involved with cytoskeleton. So it could easily be screwing up something having to do with this memory formation itself. But most of these other diseases, we don't quite know at all what the pathology is. That's why you'll also see in the New York Times someone saying, well, there's just no evidence of anything. But it's the leading hypothesis. I guess I just was concerned. In fact, if people read the New York Times, last week in the New York Times, one of the famous guys who worked on A beta, who was the head of the NIH for the Aging Institute, was found to have fabricated a lot of his results. But I think it's unlikely that those results by themselves they're not defining of the whole field. There's as much of, but it's troublesome that there are problem people there. But I think aggregation clearly is involved in the formation of structures in cells, and so I think it's interesting as physics.
Peter, curiosity, when a person has Huntington, how bad is the entire thing when you start to have the symptoms? And is there any kind of way of detecting early onset of these things?
I'm not an expert on it. I think it's a very Huntington's disease used to be called Huntington's chorea, chorea, which means that it causes people to do all kinds of difficult movements. So I don't want to do such a movement. I'm not Donald Trump. But that movement of Donald Trump where he imitates somebody with a mental illness, that's a fairly good description of what happens when you get Huntington's. You have all kinds of things that happen for your motor motions, and it's very, very sad.
But they are at this stage. They can already.
There's nothing to be done.
There is nothing to be done. The question is how early that can be detected. Is there any kind of
you can measure now with sequencing. You could find out how long your polyglutamines are now. I mean, anyone, and half the people, some people do that who have some family history, and some people choose not to do that because they don't want to be sure. And you can't be sure because you don't know whether you got the parental gene or which parent gave it to you and so on. So there's so for example, Nancy Wexler, even though she went and found this gene and became famous for that, and deservedly so, she never determined her own sequence, even until it was apparent that she had Huntington's disease. She chose not to know.
And the strategies in terms of drugs that prove.
Well, I don't know Huntington. When in the A beta project, there's a lot of interest in fully in the kinetics of the process, and there are several places where there's some weak links. So the problem is there's a fairly high concentration of protein. So the idea that you can bind to that one protein and do something is always difficult. Now you could turn the protein off, maybe, and so that's one kind of strategy to turn off the production of A beta, and I don't know how much that's been tried. The other strategy that's been in the news a lot is to find things that bind to the A beta fragments that are immune proteins to those. And the hope is that those would bind, say, it would be best if they were to bind at the tips, because if they were to bind at the tips, then you could stop the lengthening of the chains. There's another process called secondary nucleation, and secondary nucleation involves having formed a fiber and then another now forming a second fiber on the outside of that, and that's something we're starting to look at, as you know, with Murillo, actually, Murillo Sanchez back there. I don't know, do you have a poster on that? Yes, okay, he has a poster, so I'll advertise the poster.
So how different is the aggregate of Huntington compared to the other aggregates, because of all these extra hydrogen bonds that you form during the aggregation?
I don't know much about that.
Hey, Peter. About the energetics, like the A beta, when you try to break them, it's very hard, right? Like the energy to break the barrier is pretty high. Is it comparable in this polyglutamine, or it's?
I don't know the number offhand. I think we know the number, but I don't know it offhand. I mean, the graphs we always look at this as the free energy profiles at constant chemical potential or concentration. So I don't know, and you go from being uphill to downhill, more or less. So the exact number of the binding isn't, it's that number determines the concentration, but I don't remember what it gets translated into in kilocalories.
Other questions?
Yeah. Oh.
Thank you for the very nice presentation.
Okay,
It was
okay,
very cool, very nice. I think my question is more like looking forward. So you mentioned that it's very challenging addressing this type of problems because the experimental data is not super high resolution or high quality or whatever. And in cases like this, for the new generation, what would you think would be nice to have as data, experimental data, to pursue
the things that first of all,
way?
part of the complexity is stuff in systems biology. I think I had one slide very near the beginning of the various steps that go from phosphorylation down. And there's just a ton of stuff in that. When I compare that to other problems I've done with system biology, it's much more complicated. So there's a ton of that work. I think imaging is the most important thing. So I've been pushing people like our colleague Anna Karin Gustafsson to try to image the dendritic spine. But I've got to say she tried, and the problem is it's really small. Even though she's an expert in super resolution, she came from Merner's group, she actually has given up already. So I think it's very difficult. Well, maybe after she gets tenure, she'll do it.
Thanks.
But imaging, I think, is the main tool, chemically specific imaging. Yeah, Ben.
Beautiful talk, Peter. I could listen to you for hours. So I have a technical question. You mentioned sort of on the side that taking into account the concentration dependence is sort of an interesting statistical mechanical problem. I was wondering whether you can say a little more on how you address this beyond.
Well, basically, let's see, if we go back to what will take me back? It's not a super hard statistical mechanics problem. Yeah, it's not a super hard one, but basically you have to take into account that if you say take a box with 12 guys in it, that has a certain concentration. But once one molecule is already bound, then the concentration has changed from 12 to 11 in the size box, and so on. So as long as the medium outside is basically, as long as you have only one big aggregate and the other aggregates are monomers, it's a very easy calculation to just keep track of the changing concentration when you monitor N. Now it gets tricky if in fact you also form multiple aggregates. If you form things that are both, say, the big aggregate and you have some dimers and stuff like that. There still is a recipe for doing that, but now you have to keep track of how many guys are dimers and how many guys are monomers. It's quite a bit more complicated. It's so complicated that sufficiently complicated that when we've had that problem in the group, we say, let's change the concentration and simulate for longer at a lower concentration. But that can be worked out. It surprised me that almost all the simulations of aggregation that you see, there's a huge number of them, and a lot of them have important insights to them, but very rarely is the concentration specifically dealt with, or they do, say by doing peptides, you can afford to do 1,000 guys or something like that if there's a small peptide, and then you can, then the concentration doesn't change that much. So those are the sort of tricks, I think.
Thanks.
Peter, just on more technical aspects, when you run these simulations using Austin, and it's fantastic because it's very fast force field, how robust are these results when you change some of the parameters we know that there are changes that can be
Well, I mean, yeah, I don't know. We don't do a lot of exploration of parameter space because we have enough other variables to explore in these type of studies. There's in structure prediction, we've played around with that a lot. I would say I was also sort of surprised that the concentrations came out reasonably well here. In fact, when we did the A beta work to start with, we calculated the solubility. And then I asked Ming Chen, and I said, you know, can you find the solubility? That must be measured, right? And they couldn't find it. They couldn't find the solubility. And then I called up Bill Eaton, because he does all the solubility of, you know, heme and his other people in A beta. He says, oh, I don't know, but I'm sure Rob Tico knows. So then he calls up, then we call up Rob Tico or email him, and I say, what's the solubility of this? He says, oh, we've just determined it, and it came out to be the exact number we had found. So I was surprised. I was surprised that it worked that well. But we didn't have a lot of experimental data to tune in these cases. I mean, it's kind of remarkable, actually, how this sort of obviously important thing is not, yeah, this Rob Tico data would be now 10 years old or something. It's not 30 years old, right? So.
Yeah, well, we've been playing with Austin, and people sometimes get really picky that it's, well, it's a coarse-grained model, etc. But we got very nice results, and so just wondering if
well,
we are just using
when we do, for example,
stuff, so
the other kind of measure that's similar to that is the measure of the, what do we call it? Mutation stability change, right? And when that's actually fairly easy to sort of get at. I would say when you take mutation stability change using a DCA Hamiltonian, using evolutionary data, you get better agreement, but you get an R squared of about 0.7, 0.8. So again, it's not something that's going to give you always the right answer for everything. That means there's a strong correlation, but it doesn't mean that it's guaranteed to be the right number. But it's also the same as almost any other neural network type thing for predicting mutation energy changes.
perfect. Well, I think we have now our coffee break, so let's have a round of applause for Peter. Thank you so much. We have like half an hour of a coffee break upstairs, okay?